sense, that is the taste, the ability to eat, to drink something, and taste the flavor of what we are eating, what we are drinking. This, the sense of taste is closely linked to olfaction. I mean, when we have food, when we have drink, it's easy to connect the taste of this food with its smell. And because the two senses are strictly connected, they are close to each other in our body, and allow us to better understand the external environment, to understand what we are introducing in our body. Taste is a kind of complex sensation because we have the ability to taste 5 different sensations. This is a combination of 5 different sensations. When we eat food, we can taste sweet, we can taste if they are acid, we can taste the salty, the bitter, and the umami. Umami is related to the amino acid glutamate. That's also why when we have some food, if you read, there is usually the addition of some glutamate in some kind of food because it supports our taste. It supports the, it activates this the umami taste. So that's why there is the glutamate addition in some food. To taste these different, to activate this different sensation and allow us to taste what we are introducing in our mouth, the specific portion of the tongue involved in this sensation are the taste buds. Taste buds located on the tongues, and on these taste buds we have the presence of specific cell types. Each cell type regulates a specific sensation, and it is thanks to these cell types located on the taste buds on our tongue that we are able to taste what we are introducing in our mouth. So talking about the cells type, we have the type 1 cells. They are kind of glial cells. They probably allow us to taste the salt. I'm saying probably because it is not clearly verified yet, clearly demonstrated yet, that it's thanks to these type 1 cells that we are able to taste the salt. The type 2 cells or receptor cells that allow us to taste sweet, bitter, umami taste is thanks to the activation of a G protein coupled receptor, that is the gastosine, a specific receptor located on these cells. The type 3 or presynaptic cells for the sour taste. And we then have these type 4 basal cells that are some kind of precursor cells of this receptor cells. So basically the main type of cells we have and that allow us to taste sweet, bitter, umami are the type 2 cells, and then the type 3 or presynaptic cells for the sour taste. There is a probably involvement of the type 4, the type 1 cells, for the salt taste. Look at how the taste, how we are able to receive, to feel the taste of food in our mouth. We are going deeper in the analysis of the taste bud structures. So this is the taste bud location on the tongue. They are located in here on the dorsal surface. If you look the enlargement of the taste buds, you can see that located on the taste buds there are some particular type of cells that we have already introduced, that thanks to the receptors, G protein coupled receptor or ion channel, are able to interact with molecules, with substances that we want to taste, that first of all must dissolve in the saliva. And then, once they have dissolved, these ligands interact with receptors located on these specific cells. Looking at the structure, the localization of these specific cells on the taste buds, you can say that we have the type 1 support cells for salt, probably, that are located in the two lateral portion of the taste buds. And then inside we have the presynaptic cell, the type 3 cells, for in here for the sour taste. Close to the sour taste we have the receptor cells or type 2 cells for bitter, umami and sweet taste. So once you have the interaction between the ligand and its receptor located on the cell surface, this interaction is able to activate a signal cascade. The signal cascade so works activating the primary sensory neurons, that is gustatory neurons, and the axon of these neurons are basically the cranial nerves. We have the involvement of these three different cranial nerves, 7, 9, and 10. This cranial nerve synapses in the medulla, and then from the medulla, the information moves through the thalamus and then to the gustative cortex. In this case, we have the thalamus involvement. When we mentioned the olfactory system, we said that the information from the olfactory tract moved straight away to the olfactory cortex. In this case, from the medulla, information passes through the thalamus and then they arrive into the gustative cortex for the elaboration. In general, the sensory pathways move through the thalamus. The only exception is for the olfactory system. Here is how the ligands interact with a receptor and activate the signal cascade. For what concerns the sweet, umami or bitter taste, the ligand interact, the binding between the ligand and its receptor, that is a G protein coupled receptor, a particular G protein, that is the gasteusin, is able to activate the signal cascade. This signal cascade works mainly through the increase on the calcium concentration. So once we have the increase in calcium into the cell, this signal is able to support the exocytosis of vesicles that contains neurotransmitters. So neurotransmitters are released, as oral ATP can also be released. Once the neurotransmitter is released, then this signal is able to activate the primary sensory neuron, the primary gustatory neurons, that thanks to the action potential send the information to the brain. So the calcium concentration also works on the presynaptic cells that is activated. Keep in mind that the difference between the two cells, the type 2 and type 3 cells, is that for the activation of presynaptic cell, the activation of the receptor that is activated by the signal is an ion channel. So in this case, it's the concentration of hydrogenous as well as the pH change that is able to activate the signal that still works through the calcium, an increase in the calcium level inside the cells, that once again support the exocytosis of vesicles, the release of serotonin that activate the primary gustatory neurons. As we said for the sweet, umami, or bitter ligand, the activation moves through a G protein coupled receptor. The G protein, as you can see from this table, the G protein coupled receptor family is different according to the different days. That means
Sorry, Professor. Hello, we cannot hear you.
Guys, can you hear me now? Okay, so it was something great. Sorry, I didn't see the chat. Okay. Okay, so let's. Yes. Everything is working. Okay. Perfect. So let's restart, guys, talking about the ear, because I don't know if you have hear me. So I was saying that the ear is composed by three different regions: the external, the middle, and the inner ear. Okay, external, middle, and inner ear. As you can see, this is the external portion. External portion, that is this region that surrounds our internal ear, and it's the only visible portion from outside. It is important because it helps to direct all the sound waves into the ear canal. That is this structure that you can see in here. At the end of the ear canal, you have the tympanic membrane. This is a membrane that starts to vibrate when a sound wave arrives. This vibration is then transferred from the tympanic membrane to the middle ear to these three bones that we have: malleus, incus, and stapes. So the vibration moves from the tympanic membrane to the three bones, and at this point it arrives at the inner ear. What changes at this point is that the ear is not filled by air anymore, but by fluid. The point is the closure point, the point that separates these two structure, is this oval window that is connected with the stapes. These bones from the middle ear. At this point, we have the inner part of the ear that is going to be filled with fluid. In the inner ear, we can distinguish two main components, two main structures that have different functions. One is the vestibular apparatus, together with its semicircular canals, that is involved into the equilibrium. So the ability to maintain the equilibrium, to be oriented in the space, is a function of the vestibular apparatus. For what concerns the proper hearing function, the inner part of our ear that supports this function is the cochlea, this round structure in ear. We are going to see how the cochlea works, the main component of the cochlea, and how information moves from this structure to our brain for the elaboration. What is our disability? So when I'm talking and you receive the waves, the sound waves, what our brain elaborate is the perception of the energy that comes from the sound waves that you are receiving from my voice. I'm talking, you receive these waves, and your brain is able to elaborate these sounds. The perception of this sound is what you are hearing from this conversation. And basically, we're talking about a wave. We're talking about sound waves. So the characteristics of the waves are this: the frequency, the amplitude that represent the intensity of the wave, the intensity of my voice, that is measured in decibel. So the sound is the brain's interpretation of the frequency, the amplitude, and the duration of sound waves that reach the ear. This is the sound. What our brain is able to understand the interpretation of the main property of the sound waves. These are sound waves. And the characteristic of sound waves are the same characteristic of all the waves. So we have the frequency that is increasing in this picture compared to this first picture, the wavelength, so the distance between two peaks, the amplitude, so the intensity of the sound waves, the intensity of my voice. If I increase the intensity, the amplitude, the decibel of the sound waves is increasing. So the perception of this sound that is elaborated in your brain is an increase in my voice, because you receive these waves and you elaborate these waves according to the frequency. If I start talking more, if I start talking slower, or if I start talking quicker, this is a difference in the frequency, and you are able to understand this change in the frequency, as well as the amplitude. If if I reduce my voice, if I change the intensity of my voice, you receive this difference that is measured by the amplitude. That is related to how big is the peak and the wavelength, so the distance between two peaks. Of course, we have a different sensitivity. So if I'm talking, for someone, my voice might be too high. For other people, maybe not that high. This is an individual perception. It depends on your sensitivity. So it's a subjective perception that is different between people. It depends on our sensitivity, on the sensitivity of our ear. That's why we, all of us, elaborate and interpret this sensation in a different way. because we have a different sensitivity. So once you receive now I'm talking, so you are receiving my voice. The interpretation that you have is, at the end of this conversation, is that from the sound that I am saying, the interpretation of what I'm saying is a sound according to the characteristic of my voice, the intensity, the amplitude, the frequency, and the elaboration process in your brain starts from the ear, where you receive the sound waves that are coming from my voice. So sound waves move inside the ear canal. These in blue represent the waves. Once the waves arrive at the tympanic membrane, it starts vibration. The vibration of the tympanic membrane is able to transfer the energy that comes from the sound waves to the three bones, the malleus, the incus, and the stapes, that in turn vibrate. We are here into the middle ear. Looking at the structure of the stapes, this is strictly connected, is properly attached to the oval window. At this point, the region filled with air ends, and we are moving inside the ear into the inner ear that is filled by fluid. So the vibration that came from the sound waves through the tympanic membrane, the three bones to the oval window, is now converted into a fluid waves that move inside the cochlea. This is the cochlea, the uncoiled structure of the cochlea. As you can see, the cochlea is composed by this inner portion, that is the cochlear duct, where are also located the air cells that, thanks to the vibration, are able to move and are non-neuronal sensory receptor that we have in our ear. Then we have these two ducts that surround the cochlear duct. On the top portion, you can see the vestibular duct that is connected with the tympanic duct that is below the cochlear duct. So at this point, moving into the inner ear, we have these waves, these fluid waves that moves inside the cochlea. This movement is able to change the structure of the air cells. They can move on one side or on the opposite side according to which is the stimulus. Air cells, thanks to this movement, open or close their ion channels, creating an electrical signals. Electrical signals that alters the release of neurotransmitters that activate the primary sensory neurons, creating an action potential. Action potential that travels through the cochlear nerve. We are here, so the wave moves into the cochlea. On the cochlear duct are located the air cells that received basically fill these waves, and they move their ciliated ends. At this point, the primary sensory neuron that is connected to the air cells relays neurotransmitters that create an action potential that travels through the cochlear nerve. This is the cochlear nerve that sends information to the brain, where the information are elaborated. So this is the cochlea, the uncoiled structure of the cochlea. We said that this wave, the fluid wave, travel in the cochlea. In particular, into the cochlear duct, thanks to the presence of air cells that are able to receive, to feel this movement. How is this possible? So this is possible thanks to the presence of the air cells that are located in the organ of Corti. So if you look at this picture, this hollow tube is the cochlear duct that is surrounded by vestibular duct, tympanic duct. Looking at the cochlear duct, this enlargement of the cochlear duct, you can see the presence of the air cells. Air cells that are located inside the organ of Corti. That is a component of the cochlear duct. This is the cochlear duct. A component of the cochlear duct, this blue enlarged portion, is the organ of Corti. And on the organ of Corti, we have the air cells. Air cells that are connected to the tectorial membrane. So once the fluid wave arrives and moves inside the cochlea, this wave at this point is able to change the structure of the tectorial membrane, that is a flexible tissue, and in response to the fluid wave that is arriving, the tectorial membrane move, and this movement is received by the air cells. The air cells, looking at the structure of the air cells, you can see that the top portion of the air cell is composed by these stereocilia that are linked by each other by this tape link. It means that when the air cells, when a signal arrives, when a fluid wave arrives, these cells are able to move all together in a kind of synchronized movement on one way or on the other wave according to the signal that is coming. In a kind of resting situation, we have that only a small amount of the channels are open. So the air cells are these non-neuronal receptor cells. The channel involvement in this signaling are ion channels. So in a resting situation, there is just a small amount of ion channels that are open, and you can see that neurotransmitters are released. There is the action potential in primary sensory neurons that support a kind of resting situation. When a signal is arriving, if it is an excitatory signal, it means that it is supported by the depolarization of the cells. Depolarization that is regulated by ion channels that open. So ions, cations are coming inside the cells, supporting the cell's depolarization, and then thanks to the calcium increase, neurotransmitters are releasing. In this case, there's going to be an increase in the action potential in the primary sensory neuron associated with the air cells. As you can see, all the stereocilia are moving together in one direction, supporting the action potential that is increasing. And so the action potential that travels through the signal pathway and is going to be elaborated in auditory cortex in our brain. On the opposite way, if you have an inhibitor signal, the hyperpolarization of the cells is supported by channel closed. So there is a less incoming of cations in cells, so no release of neurotransmitters. So there is no action potential, so no signal is going to be sent to our brain. So we are not hearing any kind of stimulus that is coming from outside. This is the difference. So the excitation that supports the polarization of a signal incoming that allow the movement of the stereocilia on one direction or the inhibition. So the stereocilia moves on the opposite direction. This is due to the pectoral membrane that receive the fluid wave, so that feel the changes supported by the wave that is coming into the cochlear duct, and in turn regulate the air salts movement and the signal transduction, creating the action potential for the excitation or inhibiting the action potential in case of hyperpolarization, so when we receive an inhibiting signal. So how does the auditory pathways work? Keep in mind that we have two different ears. It means that we must receive information on both the ears to help our brain to localize the source of the sound. In our ear, for what concerns the sensory system, there is no receptive field. It means that for the localization of the sound source, our brain uses a different mechanism. So it is able to calculate the timing required to the activation of the sensory neurons in both the ears, and in this way is able to discriminate the source of the sound thanks to the timing required to activate the sensory neurons. Looking at the signal pathway, the auditory pathway, so once we receive the sound waves arrive at our ear, and so they move through the external, middle, and inner ear into the cochlea. The primary auditory neurons we are here project in the medulla oblongata. We are at this level. Looking at the pathway, you can see that secondary sensory neurons from the medulla project on the ipsilateral nucleus, superior olivar nucleus, but also they project on the project to both the nuclei, the ipsilateral and to the contralateral nucleus. And this happens for signals that are coming from both the ear. So, from the left cochlea, from the left cochlear branch, these primary neurons project to the medulla, and then in turn the secondary sensory neuron project to both its ipsilateral and its contralateral nucleus. In the same way, from the right cochlea, the secondary sensory neuron project on its ipsilateral and on its contralateral superior olivar nucleus. Looking at the pathway, you can see at this point that you have two different projections that are coming, one from the ipsilateral region and the other one from the contralateral region. They both project in the midbrain, and then from the midbrain to the right thalamus, the signal arrives to the right auditory cortex. It's the same situation for the left cochlea, from sound waves that arrive to the left ear. We have both the projection, one that comes from the left ear, the other one that comes from the right medulla, and both the signal project, both the neurons project in the midbrain, and then the information moves to the left thalamus and arrive to the left auditory cortex. For what concerns the cerebellum, looking at this point at the superior olivary nucleus, we can say that some information move to the cerebellum. There are some projections to the cerebellum. that are mainly involved into the regulation of the equilibrium. So, sound is processed in this way. Information from each ears goes to both sides of the brain. Information looking moves to both right and left auditory cortex for the elaboration. There can be some problems in case of hearing loss at different level of the signal pathway. So there can be no transmission of sound through either external or middle ear, or damage to the neural pathway between ear and the cerebral cortex, a damage to the cortex itself that impaired hearing, or a damage to the structure of inner ear. In all of these cases, we are going to experience hearing loss. But keep in mind that the damage, of course, can occur at each step of the pathway with the same consequence, that is the hearing loss. So I'm just going to show you this video that summarizes the situation, how the human ear work, and then we can have a few minutes of break if you're okay, guys. So now let me see if the video works. If you hear the video, guys. Okay. So if you cannot hear the video, let me see what we can do. The microphone is activated. There was something I did last time, but I cannot remember it. Oh, no. There was something I used to do for the video, but I cannot. Anyway, so guys, the video is on the presentation. So if you download the presentation, you can see it, because now it seems that you cannot hear the video. So you can basically see it by yourself when you download the presentation, okay? And so we can have a few minutes break, and then we can restart talking about the eye and the vision. If I manage to show you this video, I will let you know. Anyway, it's on the presentation. It's just to summarize what we have said right now. Okay. Yeah, let's have 10 minutes of break and then we can start. Because the online lesson is just an opportunity to help you during this particular situation. But university, I mean, this is not an online university, so you must come in presence. This is going to help you. I mean, we can have a more direct contact if there is anything that is not working, if you want to ask something. Because I know that you are online, but I don't know how many of you are really following the lesson, or you are just online to show that you are online. I don't mind, but in the end, you are going to have the exam, to take the exam and pass the exam, to move to the other exams, to move to pathology. So just keep in mind that it's in presence, this university. And I'm coming from Bologna, other professors are coming from other cities to help the lessons. So your presence, it would support our lesson, and it would be also help for you. We are going to report this situation to Professor Tumiati and see how to fix it. Because, I mean, it's not anymore that we are in lockdown or in the red zone. So you must come in presence. Okay, so moving to the sensory physiology again, and talking about the senses, we have to mention the vision. So the eye and vision. What is the vision? So is light reflected from objects. This light is translated into a mental image. That is what we say is just the light that hits an object, and our brain is able to elaborate this light ray, and give us a mental image, give us a vision of what we have, what is surrounding us. So there are 3 steps that allow us to create this mental image, to see something that is in the environment. So first of all, light enters the eyes. We are looking at something. This light ray that reflected from the object enters our eyes. According to the structure of the eyes, we have the lens that help to focus this light ray on the retina, on the focal point of, located on the retina, to see the object on focus. We are going to see that if this light ray do not converge on the focal point on the retina, our vision is not on focus. So once this light arrives on the retina we have the photoreceptors. These are transducers that are able to convert the light energy, the energy that is coming with the light ray into an electrical signal that is elaborated by our brain. So at this point, retina, brain, the information travels from the retina to the brain, where the signal is elaborated and is giving us a visual image. So this is the eye, the structure, the external structure of the eye. So looking at its location, the eye is located into this cavity, into this bony cavity that is the orbit. This cavity is supports the eye and protects the eye. Together with this cavity, we also have the muscles that are attached to the surface of the eye, to the external portion of the eye, regulating the eye movement. So we have the bone, we then have the muscles, and then the upper and the lower eyelid that allow us to close and open the eyes. Looking inside the eye structure, this black structure is the pupil. It is an opening that widens and shrinks when papillary muscles contract to regulate the amount of light rays that are coming into the eye. The pupil is then surrounded by the iris, this ring that is a muscle structure, and then the sclera, this connective tissue that supports the structure of the iris and the pupil. So, from a cavity, from a bony structure, inside this bony structure, we have our eyes that are protected by all these components, and the eye movement is supported by muscles together with the eyelid that allow eyes to open and close. Moving into the anatomical structure of the eyes and what is basically inside, what we cannot see, we have that the eyes are composed by two different chambers. One that are basically separated by each other thanks to this lens. So, one is the anterior chamber, and is this portion that is located in front of the lens. This portion is filled with aqueous humor. We are here, this is the lens, this is the aqueous humor that is produced by a muscle structure and that support its production and cover all this region. Then we have the lens that separate the anterior chamber to the posterior, the vitreous chamber, that is all this region that is located behind the lens. This region is filled with a matrix component that support the round shape, the shape, the structure of the eyes. And an important structure that we have into the vitreous chamber is the retina, where photoreceptors are located and where all the light rays converge to see a figure, to see a picture, to see an image on focus. The light enters the eyes through the pupils. The pupils is able to modulate the amount of light that is coming thanks to its movement. The pupil can change its size In this way, the amount of light rays that are entering the eyes can be modulated and help the light to arrive to the retina, and all the transduction signal is activated at this point. Because as we said, all the signals that are coming are then converted and then transduced into an electrical signal, into a conduction potential that is going to be elaborated by our brain. And it's the same for the light. So the light energy that is coming when a light ray enters our eyes must be converted into electrical signals that our brain is able to elaborate. So, looking briefly at the pathway for vision, how this pathway works. First of all, the light is coming. We have two eyes, so the light is coming through both the eyes. Once the light is coming from the pupils, then it moves through the lens to be to converge to the retina. At this point, from the retina, we have the formation of the optic nerve. The optic nerve, so we are at this point, this is the retina. The optic nerve is this structure that leaves the eye and move to the brain. At this point, that is the optic chiasm. So the optic nerve moves from the eye to the brain through the optic chiasm. And at this point, from the optic chiasm, some fibers cross to the opposite side. So what is coming from one eye, you can also look at the system from this picture, move to the opposite side, is elaborated to the opposite side of the brain. We are at this point from the optic chiasm to the optic tract. At this point, these fibers that are coming and are moving from the optic tract synapses the thalamus, the lateral geniculate body in the thalamus. From the thalamus, the information are sent to the visual cortex that is located on the occipital lobe. So from the eyes, the light ray enters the eyes from the pupils. The lens is able to change the direction and allow this ray to converge on the retina, on the focal point on the retina, to have the image on focus, and then thanks to the phototransduction, the information are sent to the brain, moving through the optic nerve, and then at the optic chiasm level, some fiber cross to the opposite side, and moving to the optic tract, they arrive at the lateral geniculate body. At this point, information are going to move to the occipital lobe, where the visual cortex is located for the elaboration. Looking at this point, at the thalamus, collateral pathway, we are here at the move from the thalamus to the midbrain. So thalamus, there is this pathway that moves from the thalamus to the midbrain, and then through the efferent division, and in particular the cranial nerve 3, the diameter of the pupils might be regulated to change its size. In this way, the amount of light rays that are entering the eyes can be regulated thanks to the cranial nerve 3, but that controls papillary constriction. So, of course, if the size of the pupil is reduced, we're going to have a reduced amount of light that is entering the eyes. On the opposite way, if the size is increasing, more light is entering the eyes. The pathway is this we have previously described from both the eyes. We're going to see that if we are using both the eyes, we are going to have a 3D vision or a 2D vision if we are using only one eye, if our vision is limited to one eye. So we said that light enters the eyes through the pupils. And thanks to the lens, these light rays that are entering the eyes are directed to the retina, where the focal point is located. So, light rays converge to the focal point that must be located on the retina to allow us to see something on focus. The distance from the center of lens to the focal point is called focal length. Once light enters the eyes, this light can be refracted or bent at two levels, cornea and lens. First of all, the light enters and moves through the cornea and then through the lens. Light can be refracted. The light refraction is influenced by some different situations. First of all, the difference in density of two media. What means that if light is passing from the air to water, the density of the two media is the density between the two media is different. So this situation, this condition influences the refraction, or also the angle at which the light rays meet the surface of the medium. The surface of the medium within the light is moving. The light moves through the lens. The lens is modified. This modification of the lens helps to converge all the light rays on the focal point on the retina. Otherwise, we cannot see the image on focus. The process by which the eye adjusts lens shape is the accommodation. This is an important process that keeps objects in focus. If the accommodation process is impaired, if there is something that is not working properly and the focal point is not inside into the retina, objects are not on focus. We can adjust this problem wearing glass. We can adjust using a specific lens that have a specific shape that support our vision. This is how light moves through a surface and arrive to the focal point. So we can distinguish two different shapes of lens. A concave lens, that is this first situation, and a convex lens. When light rays move through a concave lens, they are refracted. If we have a convex lens, you still have some parallel light rays, but they converge on a focal point. This convex lens, through light rays that move through the convex lens, converge on the focal point. If the focal point is located, like this situation, on the retina, the object is on focus. We can see an object on focus. But we can also have some different situation. In this case, if the object we are seeing is too close to the eyes, I'm putting my hand close to my eyes. If you put your hand close to your eyes, it is difficult to see the finger, to see your hand perfectly on focus. In this situation, indeed, the focal length, so the point where all the light rays converge, is out of the retina, is behind the retina. The focal length is still the same. Look at the focal length in this picture. The focal length in this picture is comparable to the C1, but the distance is increased. I mean, the point is that the object is seen out of focus because the light rays converge on a point that is behind the retina. In this situation, the object is not on focus. So if you want to keep this object on focus, the lens must change its shape. You still have the object at the same point. If you look at the D and the E picture, the distance between the object and the lens is still the same. What is changing in this E situation is the shape of the lens. The lens is becoming more rounded. Thanks to this change in the shape, thanks to this accommodation of the lens, the light rays are going to converge once again on a focal point located on the retina. In this case, the object is still in focus. So if you want to see an object in focus, you must have that all the light rays converge on the focal point on the retina. The accommodation of the lens, so when you have a change in the shape of your lens, is a process through which eyes try to see the object on focus. In this way, the light ray can converge on the focal point. The focal point is going to be located again on the retina, and so you can see your object perfectly on focus. This is the accommodation process, and it is really important to allow us to have a proper vision. The accommodation process, so these changes in the shape of the lens, are regulated by some muscle, by this structure that is the ciliary muscle that is connected to the lens using these ligaments. So, ligaments, the movement of the ligaments that is regulated by ciliary muscles that can be relaxed or contract, modulate the shape of the lens. Once the ciliary muscle relaxed, you can see that the ligaments pull on and the lens is more flattened compared to this other situation when ciliary muscle contracts. In this case, the accommodation, the change in the shape of the lens supports our vision, help us to see the objects in focus. And it is thanks to the ciliary muscles that we are able to change the shape of the lens to support the accommodation process. Ciliary muscle that regulates the ligaments structure and in turn the lens shape. Of course, there can be some problems, some vision defects that are related to problems in the retina, in the focal point localization, problems related to the lens accommodation, and is the presbyopia situation. So the accommodation, the changes in the shape of the lens is impaired. In this case, our eyes are not able to change the shape of the lens and to help us to see the object in focus. This is the loss of accommodation, and the defect is the presbyopia. We can also have two opposite problems, the hyperopia and the myopia. In this first case, the focal point is here. This yes, is located behind the retina. So basically, what is the problem? The problem is that the shape of the lens, the problem can be corrected changing the shape of the lens, and in particular adding a corrected lens. In case of hyperopia, the correction can be made using a convex lens. So if you have a convex lens, like this situation, that allow the light ray to converge into the focal point, in this case, you can correct this problem. The opposite situation is the myopia. In this case, you can see that the focal point is in front of the retina, and once again, the object is not on focus. To correct this problem, you must wear a concave lens. So when the light ray moves through this concave lens, they can converge on the focal point located in the right position on the retina. So the correction of these problems related to the localization of the focal point can be made using specific lens with different shape, a convex lens for the hyperopia or a concave lens for the myopia. If the cornea is not a perfectly shaped dome, the other problem is the astigmatism. So you have difficulty to see the object. You can see something diffuse, a diffuse image, and it's related it's related to the cornea shape. Another problem is the lazy eyes, but in this case, the problem is related to the development of the neural network. So as you can see, vision defects may be related to different conditions, not just to the lens, not just to the shape of the lens, but also to the cornea or the development of the neural network. But in this case, we are able to correct this defect and allowing us to have a proper vision. So what we see when we look around, the light ray move into our eyes through the pupil, the lens in the retina, where photoreceptors are located, and they convert this light energy into electrical signal. What we see, the visible light for us, for human, is just this region, is in between 400 and 700 nanometers. Only this portion is the visible light for us. All these other wavelengths, all these other lights that have a different wavelength are not visible for us. So it's just a really small amount that we can see. Once this energy comes into our eyes, thanks to the photoreceptors located on the retina, we have the formation of an electrical potential, an electrical signal that is able to be elaborated in our brain. And everything takes place in the retina. So we can just quickly see the structure on the retina, and then tomorrow we can talk in detail about retina and phototransduction of the signal. This is the retinal structure. Just keep in mind that there are 5 different types of neurons. First of all, the photoreceptors. Rods and cones are the phototransducers, and with 2 different functions. Rods allow us to see basically the black and white, the monochromatic vision, they support the monochromatic vision. For the color vision, we have the cones, these other structures. Then moving through the cells from photoreceptor synapses with bipolar cells that in turn synapses with ganglion cells. So the integration of signals that comes from photoreceptor takes place thanks to bipolar and ganglion cells. We then have two other types of cells in between photoreceptors and bipolar cells. We have the horizontal cells, and between ganglion cells and bipolar cells, the amacrine cells. Photoreceptors are embedded into the pigment epithelium. This pigment epithelium absorbs all the light rays that escape the photoreceptors. So once the light ray enters the eyes, the signal transduction takes place at the thanks to the photoreceptors. If some light rays are not captured by the photoreceptors, so they escape the photoreceptors, they are absorbed at this point, because if they are not absorbed, if they escape the phototransduction process, they can give us a distortion of the visual image. They can give us not a proper image, not a proper object on focus. To avoid this problem, the pigment epithelium takes all the light rays that are not transduced into electrical signal. And this is the situation. So sensory information that are coming. So light rays converge on the focal point on the retina. This information passes from the photoreceptor to bipolar neuron. Photoreceptors, rods and cones, that synapses with bipolar cells, and then ganglion cells. The axon of ganglion cells that form the optic nerve leaves the eyes at the optic disc. At this point, this fiber moved to the optic chiasm in the brain. Here, some fiber cross to the opposite side, moving through the thalamus. They arrive to the visual cortex that is located on the occipital lobe, where information is elaborated. So we can talk about phototransduction and photoreceptors tomorrow, and see better in detail how the visual pathway works. Okay, so do you have any question? Was everything clear? Okay. So if you have any question, guys, I mean, you can also email me if you want to ask anything, just let me know. I'm based in Bologna, but I come here 2-3 times a week. So if you want to talk in person, or if there is anything you want to ask, just let me know and we can arrange a meeting or anything you need. Okay, so I'll see you tomorrow, guys. We have 3 hours tomorrow. Okay.